Hi everyone, I'm Natalie and today I will be presenting to you a special type of mollusk glue called Byssal threads. Specifically, I will be discussing the Byssal threads in mussels and their biomedical applications. So this is an outline of what I will be discussing in this presentation. First, I will go over a brief background pertaining to the structure and generation process of Byssal threads. Then I will go over its biological function in mussels, biomedical applications, future research directions, and then end off with a summary of this entire presentation. Okay, so first off, for some background, what are Byssal threads? They're proteinous thread bundles that mussels use to attach to substrates. These threads are unique to class Bivalvia and are hypothesized to have evolved from extracellular matrix due to the presence of proteins resembling collagen, integrins, and epidermal growth factor. They are also shock absorbent, water resistant, and pH sensitive, making them well adapted to withstanding the crashing waves and strong winds that mussels endure in their epibenthic lifestyles. This also makes them useful in industrial applications. So now I will be going over the structure of Byssal threads. Byssal threads are around 2 to 6 centimeters in length and consist of 4 parts: an adhesive plaque, distal and proximal sections that make up the fibrous core, and the cuticle layers surrounding the entire thread. The adhesive plaque consists of special adhesive muscle foot proteins known as MFPs and DOPA, a compound that mediates adherence to wet substrates. The distal and proximal sections make up the fibrous core, and this contains elastic and shock-absorbing collagen proteins known as precols. The cuticle is a hard, extensive protective covering for the thread, made up of MFP1 and metal L-DOPA complexes, which mediate adhesion and cohesion to wet substrates, as I will discuss in the next section. The generation process of Byssal threads takes around 30 seconds to 8 minutes per thread and occurs within the ventral groove. It gets assembled by a variety of proteins. Threads are replaced every 25 days because they eventually weaken. The process begins with the mussel foot encountering and attaching to a surface using negative pressure and suction. It then forms a reaction chamber in this region of contact with the surface. It will have conditions such as low pH, low ionic strength, and high reduction to facilitate protein reactions. And at this acidic pH, muscle foot proteins have a high DOPA content which facilitates strong surface adhesion with weak cohesion. So while the foot is adhered to the substrate, liquid proteins are being secreted into this reaction chamber from 3 gland reservoirs. So as you can see, these are the 3 gland reservoirs. The adhesive proteins MFP2 to 6 from the phenol gland, so right here, are released to form the adhesive plaque. Precols are released from the collagen gland to make the core. Finally, as the thread is disengaging from the groove, proteins from the accessory gland are coating the entire thread to form the cuticle. So in this diagram, you can see that the foot is detaching. So as the foot is detaching, the plaque and thread are exposed to seawater. And upon contact with seawater, the plaque and the thread are solidified and cohere. So seawater has a high pH and iron content, which promotes DOPA iron complexation, which leads to strong cohesion between the muscle foot proteins and weak adhesion between the MFPs and the surface. So since the Byssal thread building process has been completed, strong surface adhesion isn't necessary. It's important to note that the exact molecular mechanisms underlying adhesion of Byssal threads substrates are not fully understood, but so far researchers are focusing on investigating the specific interactions between DOPA-containing proteins and the substrate. Biological functions of Byssal threads. Byssal threads play a role in mussels for their dispersal, settlement, and attachment in their sessile lifestyle. So during the pedivillager larval stage, which I have in this figure right here, the mussel larvae develop a foot. So their foot can be triggered by small currents to secrete a Byssal filament to allow them to be carried along with the current in a process termed Byssal drifting. Byssal threads can then be used to anchor onto substrates to withstand strong currents and wind. Byssal threads are also used by mussels in predation avoidance. They are used for attachment to substrates to prevent them from easily being pried off and eaten by predators such as gastropods and arthropods. They are also observed in one study to selectively trap predators, in which they are shown to secrete a greater number of threads in trapping predatory dog whelks compared to herbivorous wrinkles. So you can see in this figure I have here that mussels are trapping dog whelks using their Byssal threads. They also secrete more threads when exposed to the scent of crabs in another study. This makes sense because greater thread secretion makes it both more challenging for mussels to be pried off from substrates, as well as more difficult for ensnared predators to escape. So this might suggest some sort of adaptation or survival strategy. Next, we will be discussing biomedical applications of Byssal threads. So first off, mussel-inspired artificial tendons. As I discussed earlier, the collagens, or precols from the Byssal thread core, are elastic and tough. So the ones here. They're actually 5 times more extensive and tougher than Achilles tendons. This makes them an attractive model for surgical grafting, and so they've been studied and mimicked in artificial tendons. Another biomedical application of Byssal threads is in surgical adhesives and wound dressings. Many surgical adhesives do not function well in aqueous environments due to water reabsorption and swelling, which then weakens the surgical adhesive. The DOPA complexes, however, from Byssal threads, have the impressive ability to adhere and remain robust even in aqueous environments. This overall increases wound healing and tissue regeneration compared with sutures, which in contrast encounter difficulty in wet surface adherence leading to blood leakage. So the chemical complexes in Byssal threads have also been studied for their potential application in drug delivery systems due to their water-resistant, adhesive, and pH-responsive properties. So mussel-inspired protein nanoparticles have also been designed as intracellular drug vehicles in anti-cancer drugs due to its sensitivity in pH. Anti-cancer drugs require a release at acidic pH for effective cytotoxicity in cancer cells. Mussel-inspired protein nanoparticles make use of the iron DOPA complexes that function in cross-linking Byssal threads, as I discussed earlier. These iron DOPA complexes can effectively change their stoichiometric structure with acidic pH, allowing for the drug to be effectively uptaken by cancer cells and killed. Overall, mussel-inspired proteins have the potential to be used in more drug delivery systems for drug and gene therapies. Future research directions. Most studies in industrial applications focus on DOPA and mussel adhesive proteins. However, there are a lot more proteins and chemical complexities surrounding Byssal threads and their adhesion that remains to be elucidated. This includes how mussels regulate the chemical complexes and adherence abilities of Byssal threads in different environments, how they alter their Byssal threads to cope with environmental changes such as climate change. So the evolution of mussel Byssal threads in their composition and maintenance of adherence and robustness in response to changing environments could also be studied. So in conclusion, today I talked about Byssal threads and how they are a natural, water-resistant, shock-absorbing, and pH-sensitive adhesives secreted by mussels. They are used in attaching to substrates, dispersal, and catching predators. Iron DOPA and precol complexes of Byssal threads have inspired many bioadhesives and biomedical alternatives in the biomedical industry, such as artificial tendons, wound dressings, and drug delivery vehicles. There are still many unknowns and questions surrounding Byssal threads and their special properties that remain to be discovered. So thank you for listening to my presentation.